blueprint of life because it provides directions on how to make proteins that the organism needs, and it is also known as deoxyribonucleic acid.
DNA has an important role in protein synthesis. DNA is found in all living organisms and is a double helix made of two strands of nucleotides. Each of these nucleotides contains deoxyribose sugar, a phosphate, and a base. RNA, an equally important nucleic acid in this process, contains ribose sugar, a phosphate group, and a base. RNA only has a single strand of nucleotides, and instead of thymine, contains uracil.
When it comes to bases, there are 4 in DNA and 4 in RNA. The bases in DNA are adenine, thymine, guanine, and cytosine. The bases in RNA are adenine, uracil, guanine, and cytosine. In RNA, uracil replaces thymine.
In DNA, guanine pairs with cytosine, adenine pairs with thymine, and in RNA, adenine pairs with uracil, and guanine pairs with cytosine. In our models, dark green represents guanine, light green represents cytosine, orange represents adenine, pink represents uracil, and red represents thymine. RNA polymerase is an enzyme that binds to DNA and separates the DNA strands during transcription.
The first step of protein synthesis is transcription. In transcription, the code contained by the DNA is rewritten onto the mRNA, aka the messenger RNA. First, the RNA polymerase unwinds the DNA. It then copies the template strand's genetic information for making protein into the form of a messenger RNA. The messenger RNA leaves the nucleus. Once it leaves the nucleus, it makes its way to the ribosomes for the next and last step of protein synthesis.
In our model, thus, RNA polymerase can be seen unzipping the DNA double helix, hopping down the DNA's genetic code. Once the mRNA strand has successfully exited the nucleus, the next steps take place. In translation, the message from RNA is taken and used to build protein. The mRNA attaches to the ribosome. Then the tRNA brings the correct anticodons to pair with the codons. The ribosome then connects the codons with the anticodons, allowing for the correct amino acid sequence. 3 bases in mRNA are called a codon, and 3 bases in tRNA are called an anticodon. Proteins can be 2 to 2,000 amino acids, allowing them to vary in sequence and size. This is why there are so many different types of DNA and why DNA genes vary.